Hello friends, welcome to my channel. Today in biochemistry lecture series, I will be discussing the DNA structure. In 1953, Watson and Crick discovered the double-stranded DNA structure called bDNA. They used the extra diffraction data provided by Wilkins and Franklin, for which Watson, Crick and Wilkins got Nobel Prize in 1962 for medicine. Okay, so the double-stranded DNA, which is the bDNA discovered by Watson and Crick, is this. What is this? The double-stranded DNA or bDNA has a diameter of 20 Armstrong or 2 nanometer. The double-stranded DNA consists of two polynucleotide strands which are rich in purines and pyrimidines. They have series of purines and pyrimidines, which are the nucleotides. What are purines? Purines involve adenine and guanine, whereas the pyrimidines include cytosine and thymine in case of DNA. We know that adenine base pairs with thymine with the help of 2 hydrogen bonds, whereas guanine base pairs with cytosine with the help of 3 hydrogen bonds. Now here we can see in orange are the hydrogen bonds. So suppose this is adenine, then this is thymine. If this is guanine, then this is cytosine. So the bases are actually placed towards the inner side of the double-stranded DNA, and they are connected with the help of hydrogen bonds. These hydrogen bonds are stacked one upon another, which results in the stacking interaction. This helps in stabilizing the DNA. Now this B form of DNA is 20 Armstrong in diameter. Due to the presence of the hydrogen bonds that are not completely parallel, rather slightly tilted, results in groups during the formation of helix. So there are two types of groups in the DNA or B form of DNA. This is the major groove and this is the minor groove. Okay, so from one major groove to the next major groove constitutes 1 ton of DNA. This 1 ton has a pitch of 33.2 Armstrong or 3.32 nanometer. Okay, so the one complete ton of bDNA, that is from one major group to another or from one minor group to the next minor group, has 33.2 Armstrong of pitch and consists of around 10 base pair. Okay, so from one major group to the next there are 10 base pairs. So what is the rise between the two base pair? We can calculate from here. 33.2 divided by 10, that is 3.32 Armstrong is the distance between two base pairs. Okay, so the bDNA has 20 Armstrong in diameter, has major groups and minor groups. The two groups are separated by 1 ton, which consists of 33.2 Armstrong of pitch and has around 10 base pairs. The DNA or the double-stranded bDNA is right-handed. So the helix is right-handed helix. This is bDNA. So bDNA has right-handed helical structure. It is found most common in mammals. So it is the most common form of DNA found in natural condition. There are two other forms of DNA, that are aDNA and Z DNA. Okay, this Z DNA has a diameter of 18 Armstrong, whereas B DNA had 20 Armstrong of diameter. It has 12 base pair per turn. It is left-handed helical, so the turn of the helix is left-handed. Okay, the Z DNA is not actually found in natural condition, rather they are usually found in artificial condition. And one turn has 48.6 Armstrong of pitch. So the Z DNA is much longer per turn but much narrower than B DNA, whereas B DNA is 20 Armstrong in diameter but the Z DNA is 18 Armstrong in diameter. It is left-handed, whereas B DNA is right-handed, and it is not, that is Z DNA is not naturally found usually. Okay, so these are the double-stranded DNA. Now there are two other forms of DNA. These are triplex DNA and quadruplex DNA. Okay, so the triplex form of DNA. Triplex DNA, from the name it is quite evident that triplex means that the DNA has triple strands or 3 strands. Okay, so in addition to the 2 strands, there is one extra strand that is wrapped around one of the 2 strands. Usually the third strand is rich in pyrimidine or polypyrimidine, whereas the second strand is rich in purine or polypurine. Okay, the third strand base pairs with the second strand through Hooke's-Steen base pairing scheme. Okay, so if the third strand is rich in pyrimidine and the second strand is rich in purine, then the base pairing is Hooke's-Steen base pairing. On the other hand, if the third strand is rich in purine and the second strand is rich in pyrimidine, then the base pairing will be reverse Hooke's-Steen base pairing. Okay, in case of triplex DNA, it is written as the first strand, suppose the first strand is DT, second strand then will be a purine or DA, and the third strand will be again a pyrimidine, but this third strand will be written in italics. So in case of triplex DNA, the strand which is written in italics will be the third strand. Similar to the triplex DNA, another type of DNA is the G quadruplex. This G quadruplex structure looks like similar to this. This G quadruplex is a structure of DNA which gives quadruplex, means 4-stranded DNA. This 4-stranded DNA is rich in guanine and looks like a cube. Okay, looks like this. So this is the structure of a G quadruplex. It is a DNA which has 4 strands which are rich in guanine. It can be parallel or antiparallel. Parallel means 4 strands are all 5' to 3' direction, and antiparallel means 2 strands will be in 5' direction while the other 2 strands will be in the 3' direction. So today we first understood the different types of DNA structures. Now let's understand the stability of DNA. What factors affect DNA stability? First, hydrogen bonding. hydrogen bonding between the bases or base pairs leads to stability of DNA. More hydrogen bonding means more stable DNA. Okay, so more GC in the DNA, more hydrogen bond, because GC base pairs with 3 hydrogen bond. So more hydrogen bond means more stable DNA. This hydrogen bond leads to stacking interaction, as I have mentioned earlier. Now the stacking interaction helps in DNA stability. This stacking interaction involves two types of interactions. These are, one is the hydrophobic interaction and the other is the van der Waals interaction. These two interactions help stabilize the DNA. And finally, the presence of phosphate group on the DNA backbone. This phosphate bond present on the two strands of DNA help stabilize the DNA. So we understood the stability of DNA. Now let's finally understand the thermal denaturation of DNA. Now coming to the thermal denaturation of DNA. What is denaturation? Denaturation does not mean, in case of DNA, which is also known as melting, this melting of DNA does not mean that the DNA is disintegrated into pieces. Rather, melting or denaturation of DNA means that the double-stranded DNA is dissolved into single-stranded DNA. Here we can see this is a double-stranded DNA, dsDNA. After temperature, that is due to thermal denaturation, where temperature is used to denature the DNA, after certain amount of temperature, the DNA is suppose denatured like this. So this part of the DNA has been denatured. Okay, so DNA denaturation does not mean that the DNA is disintegrated into pieces. Rather, DNA denaturation or DNA melting means that the double-stranded DNA is melted into single-stranded DNA. Now, with the increase in denaturation, also leads to the increase in absorbance. Why? Because the nucleotides consist of aromatic bases in DNA. The aromatic bases, when it is present in the double-stranded structure, are stacked towards the inner side of the DNA. As a result, they cannot absorb the UV light. When the DNA denatures, the aromatic compounds or the aromatic amino acid aromatic bases can now interact with the UV light and absorb. As they absorb more and more UV light, the absorbance also increases. This absorbance is at 260 nanometer UV. Okay, so with the increase in denaturation, the absorbance of the DNA also increases. So from studying the absorbance of DNA, we can understand how much the DNA has been denatured. At maximum absorbance, the DNA has been completely denatured. Okay, now comes the term melting temperature. This melting temperature is expressed as Tm. What is Tm? Tm or melting temperature is the temperature at which a DNA molecule has been denatured half, or half of the DNA has been denatured. Suppose a DNA is 2 meter long. So when the DNA is denatured 1 meter, the temperature at which the DNA has been denatured 1 meter or half of the complete DNA is called the melting temperature. Okay, so the melting temperature we can understand from this graph. Okay, so this side is the absorbance at 260 nanometer. This side is the temperature. We will get a curve which looks like this, that is a sigmoid curve. Why sigmoid curve? Because at the start or during the initial stages the rate of DNA denaturation is high, then the rate of DNA denaturation slows and finally ceases. Okay, so this gives a S-shaped or sigmoid curve. Now this S-shaped sigmoid curve, suppose this is the point which is the half of the maximum absorbance. So when the DNA will be completely denatured, the absorbance that will be, it is the half of that absorbance. So this is the point where the DNA denaturation has been half, or half of the DNA has been denatured. So at the temperature where DNA has been denatured half, or half of the DNA length has been denatured, suppose this point is called the melting temperature or Tm. Okay, so melting temperature is the temperature at which the DNA has been denatured half. Clear? So half of the DNA is denatured at which temperature? At the melting temperature. Now this melting temperature depends on some factors. What are these factors? One is the GC content. Okay, GC content. What happens? More GC means more hydrogen bonds, as GC has 3 hydrogen bond, whereas AT has only 2 hydrogen bonds. So more GC means more hydrogen bonds. More hydrogen bond means more temperature is needed to break the double stranded DNA molecule. So more GC content means more melting temperature. Okay, in addition to GC content, ionic strength and pH of DNA solution also affects Tm. Okay, so today I discussed the DNA structure, mainly bDNA and also zDNA, triplex DNA, quadruplex DNA. In addition to that, we also understood the stability of DNA, the factors that affect the DNA stability, and also what is thermal denaturation and melting temperature or Tm. Okay, if you have any doubt about this video, please leave comment in the comment section, and also please like and share the video and subscribe my channel Study with Snehasis and press the bell icon to get regular notifications. Thank you.